Okay, let's talk about movement of these electrical signals across a neuron. Sorry. First we're going to talk about passive spread of depolarization. So remember, depolarization is when the membrane is becoming more positive. We also talked about the dendrites as receiving the signals. So as a dendrite receives the signal, what you see across the cell body is kind of like this ripple. So if you've looked at water and ripples spreading out from the source, so you get this passive spread of depolarization. So the membrane's becoming more and more, or the membrane is becoming positive, and we're going to talk about it reaching that axon of Hillock. But it's passive spread. It's slow, and it can lose magnitude or strength as the electrical signal is going across the cell body. And that is because the sodium voltage gated ion channels, right, the ones that get this whole action potential going, are sparse or diffuse, not a lot across the membrane of the cell body, right? So you get this movement of depolarization, right? But it's not very strong, and just like a ripple moving, it can fade out. At the axon of Hillock, or the axon Hillock, sorry, this is where there's a large concentration or large number of sodium voltage gated ion channels. So now all of a sudden you can get these opening, and you can get this increase in positive charge, and that's going to reach past threshold, and then we're going to talk about active propagation of the signal. So passive spread just means that the positive charge is moving. We've got some sodium voltage gated ion channels that are opening, but we're not really getting past that threshold level until we reach the axon Hillock, where it has a large concentration of sodium channels, and here is where we initiate the action potential. So we've hit the axon Hillock. We are now propagating. So now it's actively generating action potentials. Ooh. Action potential. Okay, so now we're going to get that high level of depolarization and the whole cycle of repolarization. And I want you to notice that the action potential propagation is shown in little areas. It's not this spread like across the cell body. And each of these little areas represents a node of Ranvier. So now what's happening is the action potential is going from spot to spot to spot and getting reinitiated at each time, because at the nodes of Ranvier there's a high concentration of sodium channels, or of sodium voltage gated ion channels. So to get an action potential to be able to move all the way down an axon, it has to be propagated or continually reinitiated. So the first place it starts is the axon Hillock, and then it is reinitiated at each of the nodes of Ranvier. So this signal does not fade, it's generated anew, it actually moves a lot faster than the passive spread. So again, this picture of the myelin sheath and the nodes of Ranvier. Remember that the myelin sheath is discontinuous, right? So we have this space in between. Remember again that the myelin sheath is important insulator, so it stops the membrane from retaining charge. And instead that charge flows here where there is lots of sodium voltage-gated ion channels, and we get a new action potential that can travel to the next. So it's kind of like the action potential is jumping from node to node to node. And those are just one more picture. The reason I showed this is I like to help you understand that here in the node is where the sodium channels are. Underneath the myelin sheath is where the potassium voltage-gated ion channels are. Okay. So here we get the initiation of the action potential. You know that you eventually need the potassium channels to open up to get repolarization, and so this kind of illustrates how they're all organized. What's also really interesting is the myelin sheath, how it interacts, and it's always hard when it's 2D, not 3D, but it interacts tightly here with the membrane of the axon, and in fact blocks these voltage-gated ion channels from moving. So we talked early in the semester about the fluid mosaic model. Well, this is a spot where there is no fluid mosaic model. There's actually interference here to keep these sodium channels concentrated here and potassium over there. Just making sure we got everything. Okay, this is called saltatory propagation. And this diagram is showing that without those nodes of Ranvier and the myelin sheath, the signal kind of fades out. Right? And I have no idea why they have to spin around, with the myelin sheath and the propagation, the signal can make it all the way to the end of the axon and moves a lot faster and stronger signal. So saltatory propagation is this idea of repropagating, hopping from node to node. This is just to illustrate what can happen when you have damage to the myelin sheath. So in multiple sclerosis, it's a disease where the our body, basically autoimmune disease where our body attacks the myelin and damages it. And so what's happening is these people are having this short conduction. You can see even the action potentials are much greater dispersed and much lower. And so you have the symptoms of multiple sclerosis where you don't have the muscle control because the nerves aren't being able to send the signals like they normally could. All right. I'm going to see if we can make it all the way to the end of this PowerPoint. Okay, synapse types we have talked about previously. So what happens when this action potential reaches that synaptic bouton? Remember, there are two types of synapses. There's the electrical synapse, where when this action potential reaches, ions just go right through those gap junctions and you propagate the signal to the next neuron. So we're not going to talk about this. We're going to focus on the chemical synapses, which involve something called a neurotransmitter. So what's happening here, remember, is there's a 20 to 55 nanometer space. And so the presynaptic neuron, the action potential is going to travel down, and it's going to trigger, and we'll talk about calcium voltage-gated ion channels in a minute, but it's going to trigger the release of these neurotransmitters, which then have to diffuse through the synaptic cleft and reach a receptor. So a receptor is a protein, transmembrane protein on the membrane that is there to receive ligands or molecules like this and then do their job. So neurotransmitters are the chemical signals that are secreted into the cleft, diffuse across, and bind specific receptors. And I'm not going to go into all the different receptors and neurotransmitters, but that's something you are responsible, so check out your objectives. Okay, what I just want to talk about is that neurotransmitters can be excitatory, which means they will stimulate an action potential in the postsynaptic neuron. So they promote depolarization. Right? That's what we need for the action potential. There are also what we call inhibitory neurotransmitters. And what they do is they inhibit the initiation of an action potential, which they do this by promoting hyperpolarization. Okay. Oops. Uh-oh. What is this? Okay. So remember, if you hyperpolarize, you get too negative, it's harder to get past that threshold. So these inhibitory neurotransmitters actually hyperpolarize the postsynaptic neuron. All right. Now my thing does not want to go. Oh my goodness gracious. I can't figure out which way to go with my... Okay, got it. Neurotransmitter receptors. So remember, these are going to be on primarily the postsynaptic neuron. You can see that they're transmembrane proteins. And we have two main types. We have ones that are called ionotropic, which means when they bind the neurotransmitter, they open and let ions through. So these are ligand-gated ion channels. And the ligand is the neurotransmitter. So when the neurotransmitter binds, the channel opens and the ions flow across. And you can imagine this results in faster action potentials because you've got movement of ions, which is going to change the membrane potential. Metabotropic means it has to kind of go through a couple more stages. So it's an indirect opening of ion channels. And in this case, it's showing a G protein coupled receptor, which you are going to learn about next chapter. And so what's happening is the receptor binds the ligand, which is the neurotransmitter, and does some cell signaling that eventually opens the ion channel. That's why it's indirect, and you can imagine that it's a slower way of creating an action potential. So how are these neurotransmitters released? Well, one thing is they are stored in synaptic vesicles. So you've learned about vesicles, right? Those little membrane spheres that transport things from the ER to the Golgi, and the Golgi to the plasma membrane and to other places. So synaptic vesicles store neurotransmitters. and they require calcium to release, I'm going to put NT, neurotransmitters, by the method of exocytosis, which you learned about. So what's happening here is you've got this synaptic vesicle full of neurotransmitters. And vesicles, you know, need the snare proteins to dock and fuse. So, in fact, the synaptic vesicles will sit right at the edge of the membrane, which is called docking. But until calcium comes through, they can't fuse. So calcium comes through, or comes into the cell, via calcium voltage gated ion channel. So see how this is all working together? We're sending the signal down, comes down to the membrane, the calcium channel opens, you get calcium in here, you get vesicle fusion, neurotransmitters are released. Very, very controlled process. So let's look at this a little bit more. So there's a protein I want you to know about, synaptotagmin. It binds calcium and is required, and synaptotagmin is represented here by this little blue Ba protein. And as this vesicle is primed or docked, until calcium comes in and binds synaptotagmin, you don't get fusion. So these are your, you can see down here, these are your snare complexes. Here, snare. And synaptotagmin must interact with the snare complex to allow the release of the neurotransmitters. And that is dependent upon calcium influx or into the cell, which is dependent on a voltage-gated calcium ion channel. So you got to get that signal all the way down to the end of the axon bouton, in order to open up and get those calcium ions in. And what you can imagine is, as you're getting all of these vesicles fusing, remember the membrane at the bouton gets larger and larger, and that's not good. So we have to recycle these vesicles to avoid increasing the membrane. And there's two main methods: kiss and run, and this clathrin-coated vesicle. So kiss and run is just a temporary fusion of the vesicle. So it releases some neurotransmitters, it reforms and recycles. Or if it's gone all the way through exocytosis and added to the membrane, then clathrin can form an endocytic vesicle and recycle the membrane right now. So this is called compensatory or compensating. How about that? Endocytosis. Okay, finish up. We got just a couple more slides here. What's the fate of these neurotransmitters? So the red little dots here are showing you neurotransmitters. They're binding the receptor, but it's not a permanent interaction. So the receptor can release the neurotransmitters back into that synaptic cleft. We need to do something, or else the cell, the postsynaptic cell, would be continuously signaled, whether it's excitatory or inhibitory, by these neurotransmitters. So we need to get rid of the neurotransmitters out of the synaptic cleft. So one method is using an enzyme and degrading the neurotransmitters, so just breaking them down. and the other is reuptake. So that means the presynaptic cell is pumping the neurotransmitter back into it so it can use it again. So regardless of the method, the goal is to clear out the space so that we can have a new signal between these two neurons. Okay. Last concept is the concept of summation. And, oops, oops, what am I doing? Previous. Well, that's not what I wanted. I'll do this, and I'll pick this other one. Okay. Maybe. Okay, summation. We have temporal and we have spatial. So I want to talk about temporal first. And the idea is, how do we reach above the threshold potential? So one action potential hitting the next neuron does not stimulate a next action potential. You actually need multiple signals. So you need multiple signals to get past the threshold value, or the threshold, I should say, potential. Okay. One way you can do it is temporal. So that means from one neuron to the next, you're just sending action potential, action potential, action potential, and eventually this reaches above threshold and the signal continues on. So it's a rapid succession of action potentials from pre to post. So temporal has to do with timing. The other method is spatial. Spatial means you're getting signals from multiple neurons. So each signal is called a PSP, or postsynaptic potential. So each one has a little bit of depolarization. If it doesn't reach threshold, you get repolarized. Then you have to try again, and you have to try again. These would be excitatory, right, because they're depolarizing. This would be inhibitory. Okay. So you can have postsynaptic potentials that are excitatory. Go back. Oh no. I don't want to stop. Okay, I think I'm on a new way to record and it doesn't stop till I tell it. Maybe I'm talking to myself. Okay. Okay. So you can have excitatory, so EPSPs, or inhibitory, IPSPs. And what this graph is trying to show you, the green are excitatory, the red, purple are inhibitory, and it's the summation, accumulation of all these signals that eventually allows you to reach past threshold and start an action potential. So the inhibitory are going to work against by hyperpolarizing. The excitatory are going to depolarize, and it's the summation at the axon hillock which allows you to initiate the action potential. Right? So remember, your axon hillock is where you're initiating the action potential. Otherwise, it's just diffusion of these small signals. Okay, so temporal means from the same neuron, rapid fire. Spatial means from different neurons. And what I just want to show you, and this is not terms that you need to know, but just appreciate that it's way more complicated than what we just went through, of course. Not all signals are transmitted from axon to dendrite, right? You can have axon boutons secreting into the bloodstream and into different regions of the axon, okay, and to different parts of the dendrite, not just always at the tips, and axon to another axon. So it can get super complicated. The concepts are the same, and for all of them you need to reach that membrane potential past the threshold, so that you need to reach the threshold potential in order to trigger an action potential. And that is it for chapter 22. I hope this helps.